everyone, and welcome to Labceutics' first international webinar. Thank you for joining us and taking the time out of your day. We know that you're all very busy. I'm Mark Reese. I'm Vice President of Business Development for Labceutics, and I'll be your moderator today. And today we're going to give you an update on Labceutics, and we're also excited to have a special guest speaker, Dr. Simon Patton, who will be discussing what you need to know about external quality assessment in today's laboratory environment. Before we start the program, just a couple of housekeeping notes. You'll notice that you are on mute, so only the speakers can be heard, but you'll see in your console there's a question box. So if you have a question during the presentation, please go ahead and type it into the console, and then we'll answer all questions at the end during the Q&A. So please feel free to type in a question at any time, and we'll answer all questions during the Q&A session. Also, this webinar is being recorded, so if you'd like to review it or share it with any of your colleagues afterwards, you'll receive a link to the recording. So with that, I'd like to introduce Tiffany Olson, and Tiffany is chairwoman and on the board of advisors at Labceutics and Diaceutics. Tiffany was most recently the Vice President and Global Head of Diagnostics for Eli Lilly and Company, where she was responsible for developing and implementing its diagnostic strategy and providing innovative solutions to enable the integration of diagnostics into pharmaceutical development for personalized medicine. Prior to that, Tiffany was the President and CEO of Roche Corporation. Tiffany?
Thank you, Mark, and welcome everyone. As Mark read my bio, I wanted to give you just a little bit more information about it and why I'm so excited about Labceutics. I've been involved in personalized medicine for about 12 years and have worked closely within both the diagnostic and the pharmaceutical industries. And you hear a lot about personalized medicine for the patient, for the diagnostic manufacturers, pharmaceutical companies, doctors, pharmacists, but you hear very little about the laboratory. And we feel that the laboratory is truly the forgotten stakeholder in the personalized medicine chain. And understanding that is one of the reasons why we started Labceutics. So our mission is to connect laboratories, diagnostic companies, and pharma to help to optimize the implementation and commercialization of companion diagnostics. So Labceutics helps to make an efficient personalized medicine network because it offers opportunities for really testing access, quality standards, and education up and down the personalized medicine chain, along with real-time communication across the stakeholders. Pharmaceutical companies benefit by really understanding their desired needs, regardless of whether it's a kit or an LDT for their very specific therapeutic launches. So they can easily communicate directly with you as you have access directly to those physicians who are prescribing both the test and the drug and helping to improve the adoption and the understanding of the testing. So what you all have told us is that keeping your academic and your scientific leadership is extremely important, along with the ability to continue to build your reputation as a premier personalized medicine laboratory. We help you by facilitating some of the best practices, the SOPs, and other information, along with aiding and helping you to get the highest return on your money for PM testing. So Labceutics is here to help you be leaders in this field. And with that, I'll turn it over to Mark, who I believe will introduce Maria.
Great. Thanks very much, Tiffany. So I'd like to introduce Maria Paz, who is the Managing Director of Labceutics. She's based in Madrid. Maria has over 10 years' experience in healthcare and biomedical industries, particularly in epigenetics and pharmacogenetics and molecular diagnostics. Prior to joining Labceutics, Maria was Head of Translational Medicine and Scientific Development at Zelchia SA, where she coordinated the international labs and hospitals and has over 7 years' experience setting up and managing laboratories for molecular diagnostic pharmacogenetics. Maria completed her studies at Johns Hopkins in the U.S. Maria?
Thank you, Mark. thank you to the audience to be here this afternoon with us in Europe. Just to put a little bit of framework around with beneath the network that we have, and as Tiffany very well explained, we started building up the network involving the different labs in EU5. Now we are expanding to other markets in other continents. But essentially, what we wanted to create was more than just a network, and to build a dynamic network. And for that, we set up a tool called what we call Connect, which is a connectivity platform, in order to allow the labs to really have that visibility that we want to promote from Labceutics. Our main mission is to involve the labs as a major stakeholder in the personalized medicine setting. So we realized that we needed to empower the labs. And in order to empower the labs, while we were promoting networking, we created Labceutics Connect as an excellent resource to put at the disposal of the partners at the same time as we were managing the different projects that we might have from the industry. So basically, it is a tool that will enable our partners to first networking and partnering among them, also to gain that visibility abroad and outside their scope, and also to promote the knowledge flow and the intel around personalized medicine. So just to start showing in the first slide, here you can see it's a snapshot of the network to give a broad view of the network as it is in EU5. Now we are including Asia, Canada, and Brazil. But this is one of the models that Connect will have for you as free of access, and I will come back to this in a minute to explain more about the tool. But essentially, this network model will allow you to identify other partners and establish any sort of communication with them, zooming in each of the flags that you can see there, either in Germany, France, Italy, wherever you go. You can click in each of those flags, and then you'll retrieve the information of each of those partners and establish a contact with them. And also at the same time, it's also a tool, like a showcase or a shop window for you to promote your lab services and your own lab portfolio, and thus being able to open it to a broader community. So this is basically the network model. Can you please go to the next slide, Mark? Thanks. So this is how the homepage looks like. There you'll see in the top right corner there is information. There are links to your own information as a user, and then to your own institutional profile, which is visible to other partners. So it's a way to gain that visibility. But there is also a link that you can also find at the bottom right there, where you can see information about the Labceutics survey, which is the information that we want to get from each of you, and it's specific information about the technical capabilities and skills of your own lab. And this information will allow us to match it with projects and figure out which is your suitability and potential interest in particular projects to fit in and to show you those projects to participate in. So that's in regards to the service and the information that we get from each of the partners. And then if you can see in the homepage there is more information, a quick overview of the latest news posted in the different sections that we have in the second model, which is the forum, apart from the network. And then there is a dashboard above that will lead you through the different sections as well, and the different models. There is a huge banner here where we want to add the main activities that we are promoting, like travel vouchers or new educational activities or initiatives. And in the right side, you can see the forum index, the network model, and then the survey model. If you can go to the next slide, I can show you a little bit more about the homepage and zoom it in to get more information about the forum model. Basically, with this model, what we want to do is to highlight the main goals of our purposes in Labceutics, which is give the opportunity to partners to participate in specific projects with leading pharmaceutical companies, and also to have that visibility that we want to ensure, and provide members with the capability to introduce and share new and innovative tests and protocols, and share reagents, share any other activities that they want to promote from their own setting, like training activities, educational activities. We want to promote discussion through the blog, the development and standardization of protocols and test procedures, sharing whatever you may want to share, like training, exchange, etc. We want to also provide regular updates and news and opportunities for laboratories to be at the forefront of this sector, and also facilitate the laboratories networking with other experts. So basically there are three branches. One is gaining visibility for your own lab. Second one is networking and partnering with other labs for scientific initiatives, funding or granting for other tasking purposes, and then the knowledge flow, information flow, and news and topics in personalized medicine. So once you log in, you'll have access to the different sections. If we can go to the next slide. This is just to quickly present you some of the top lines about the general section, and for the sake of those of you in the audience that don't have had the chance to use it, this is the general impression of the general section. There are some other sections and projects that is specific for those labs that are participating in ongoing projects. There are other sections for grants and training or partnering. But in the general section, which is one of the most interesting for lab partners, you can find subsections on accreditations with information on the general topics and general information in the accreditation in Europe, and information on ISOs and CE marking, etc. And one of the jewels of the crown of this blog is the group purchasing, which is very important and very interesting for partners, because we are in a very good position to negotiate prices and discounts for partners on behalf of a network of a huge network of labs. So we are at the moment negotiating discounts in reagents, in platforms, also in journal registrations, or in the fees and registrations for congresses, etc. But you can also find some other information about protocol development or procedure standardization, quality assurance, or other tips that might be of your interest. And what I want to highlight here is that mainly the value of this tool is the value that each of the users are willing to put in, so as to make it really valuable for you to use it regularly. So that's why we are building up the network and building up Connect to gain a critical mass that would bring more knowledge and more posts to the forum, as to make it more variable and more attractive for you. So scaling up the network to improve the appeal of Connect, what we want to ensure is that it's enticing enough to bring more comments, more user, and more knowledge flow. So this way we encourage you and invite you to invite other folks that you would like to hear and that you believe could be interested in being part of this network. And just to finalize, to go to the next slide, please. This is just to show you one of the latest initiatives that we are promoting. It's a chain of recorded videos where we invite our partners to in different tests and topics like the ALK rearrangements testing, which can be it's one of the trendiest tests that could be of your interest, and we kindly invite you to share your suggestions and comments here to us, because we are building a network, we are building Connect, and we are improving it to make it more convenient for you all. And this is all on my side. Thank you.
Great. Thanks, Maria. Really, really appreciate that. Well, I'm very happy to have the opportunity to introduce Dr. Simon Patton, who is the Executive Administrator at the European Molecular Genetics Quality Network, the EMQN. The European Molecular Genetics Services, which are essential for any laboratory seeking to maintain and provide a quality service. The organization was established in 1998 and currently offers 28 disease-specific and 3 technique-specific EQA schemes for molecular genetic testing. Simon?
Thank you very much indeed, Mark, and good afternoon to everybody, and thank you for the invitation to talk to you. Mark's kindly introduced a bit of brief background about the EMQN, and I suppose before I start, next slide please, Mark. Next slide, Mark, please. Thank you. Just a disclosure that I do work for the EMQN, as Mark kindly said, but in most of my talk today, I'm not really going to be plugging the EMQN per se. I'm really just going to be talking about the context of what EQA is, why it's important, why quality itself is important. Then I'm going to give you some examples of the improvements you can see in laboratory performance as a result of participating in the EQA. And finally, I'm going to give you a sort of overview of some of the EQA providers for the tests or the disciplines, for example, that might be relevant to the audience today. So next slide, please, Mark. So I'm just going to sort of set the environment for this talk, and really going back to a set of guidelines that were published in 2005. Originally by the for the Organisation for Economic Cooperation and Development. And they published guidelines on quality assurance for molecular genetic testing. Next, please, Mark. And if you haven't got a copy of this guidelines, I thoroughly recommend that you access it. You can access it directly from the OECD website. I'm not going to go into all the details behind it, because it's an all-encompassing document about molecular genetic testing per se in the 18 member countries of the OECD, but I'm just highlighting some of the main conclusions that came across from it. And they essentially ascertained this information from a very, very large survey that they sent out to all the member countries back in about 2003. It took about 2 years to assimilate the information before they published it as guidelines. And some of the main findings were that there was an enormous amount of exchange of samples across borders, national borders, somewhere in the region of 18,000 specimens in 2002. 64% of molecular genetic testing laboratories received specimens from other countries, and that this feature was a widespread feature of all service provision in each country. Next, please, Mark. The standards themselves are broken down into several sections. And the first section, dealing with the sort of general principles and best practices for genetic testing, highlights areas such as ethical standards, stating that testing should be part of healthcare, all tests should be quality assured, that we should be seeking informed consent and offering counselling to patients. They talk about the issues of privacy, the benefits of international cooperation, the use of the samples themselves, and I think also importantly about the advertising claims used by the laboratories themselves. Next, please, Mark. Section C deals with proficiency testing, and this is the main area of obviously my talk today. And it talks about the principles and best practices, and states that laboratories should be encouraged to participate in external quality assessment, as it sets the standards, addresses problems, and provides more information to the public about how laboratories are performing. It also talks about supporting and recognising effective and competent proficiency testing and keeping the pace with technology. And I guess with the latest developments on the testing market now, particularly next gen, that's even more important than ever. Next, please. So why quality? Why do we really need to be focusing on the quality of the service we provide? Well, I guess we ought to be thinking about producing a valid test result. We also need to be thinking about meeting the needs of our service users, particularly as we are now working in a complex national and international market. We need to be managing the risk of the service we're providing, ensuring we promote efficiency in our testing processes, encouraging innovation in the testing market, and crucially, improving the comparability between different laboratories. We need to be sure that we're all doing the same thing and getting the same result. So if we have a test in country A, we need to be sure that if we get the test in country A, the result's right, and it's the same as it would be in country B, C, or D. Next, please, Mark. So Henry Ford said that quality means doing it right when no one is looking. Next, please, Mark. Next, Mark, please. Aristotle, going back a long time before that, said quality is not an act; it's a habit. And I thought when I was thinking about quality, I was also looking. If you look at the picture in the slide, you can see that there are some workmen doing a good job of laying some new slabs, but they've made a crucial mistake. They've not been competent enough in what they're doing, and they've forgotten to allow themselves space to get their van out. So quality is not just about doing a good job, it's also ensuring that whoever's doing it is competent at what they're doing. Next, please. So back in 2003, the IPTS, the Institute for Prospective Technology Studies, published a report which I guess mirrors a lot of the findings from the OECD survey, that there are well over 1,000 laboratories and centres in different laboratory settings, testing for more than 1,000 rare diseases, and now I think we know that's at least 3 times higher than that in terms of tests that are marketed. There's a lack of centralised and uniform information about services. There's limited networking, lack of harmonised and standardised proficiency testing and EQA, a lack of reference materials, and although that's not a subject of my talk today, that's still a major issue for laboratories, the lack of reference materials. A limited number of laboratories are accredited, and the relevant ISO standard is 15189. And again, that's still a major problem even now. So back in 2003, it was highlighted again. In 2012, it's still an issue. And there are a limited number of accredited EQA schemes, although that now is improving with the new international standards for EQA schemes. There's limited analytical and clinical validation of tests, but I think this is changing, and certainly my experience is that labs are much more proactive about validating their tests properly, but the evidence still shows, particularly from the molecular pathology schemes, the sort of new personalised medicine type testing that's coming through, that's still not being done very well, and there's a very diverse regulatory environment. And all of this was highlighted in the next slide by an unacceptable number of laboratory errors being made. Next, please, Mark. So just to take you through a few of the thoughts about why genetics per se is unique in many ways in pathology. So a genetic test can be highly predictive of a person's future health. It can be carried out at any stage of their life and be applied for both prenatal or pre-implantation diagnosis. It's also highly relevant to healthy people as well as those showing disease, and may also have important implications for the relatives of the person tested. So I think in the laboratory it's really crucial that if we're doing a single laboratory test, that we need to establish where the result, the genotype itself, forms part of their permanent medical record. Next, please, Mark. We need to ensure that the test itself is correct, and the interpretation of that test, the clinical and the biological interpretation of the genotype result, is also correct. Next, please, Mark. So what is external quality assessment? Or proficiency testing, as it's called in the US, but they're essentially the same thing. It's an external process by which the standard of any assay or test is performed against a set standard. So, for example, it's usually done by essentially sending out a number of highly characterized samples to a laboratory asking them to test the sample, and then report the results back to an independent organisation. But the key point here is that the organisation that does it is independent. Next, please, Mark. Next, please. That's it. So why is EQA important? Well, partly because we're obliged under the regulations, the standards of the accreditation standards 158189, to participate regularly in EQA for each diagnostic test we perform. The OECD guidelines, which have been adopted by all of the 18 member countries, make it a specific requirement for the quality and competence of a laboratory, and state that participation in EQA is recommended. No, they don't state it's mandatory. And laboratories really, I think, have a responsibility to be ensuring that their processes are performing properly by participating in these activities. The EQA programs themselves can check the entire examination process from, the entire lab process from start to finish. And the laboratories themselves can develop a policy that includes the use of and regular participation in EQA. And as a consequence, they can address any actions that arise from the findings of EQA through their normal quality management systems. Next, please, Mark. So EQA itself has a surveillance role. So laboratories adhere to the professional guidelines and international standards that have been set by their profession. EQA itself is comparison between these laboratories, so it can help define good standards or best practice. And in many cases, EQA providers are quite involved in the production of best practice guidelines. Regular assessment of the laboratory compares its performance against a set number of standards, but also allows the lab to benchmark their performance against other laboratories. Participation in EQA is an incredibly important educational tool. It allows the laboratories to improve their overall services, and I think this is the main benefit of EQA is the educational aspect. It also allows you to validate your tests to a certain degree, because EQA materials are, although not a higher order reference material, certainly could be defined as a reference material and used in validation of laboratory tests. And I think it also helps to build public confidence in the laboratory testing process itself, if we can demonstrate that the laboratories themselves are passing the EQA and therefore doing a good job of the testing process they're doing. Next, please, Mark. So regular participation in EQA is really important. The relevant ISO standards don't actually state a time frame or define what regular is, but I think it's fair to say that I would recommend laboratories participate. I'm not saying this because I'm a provider, but I think the benefits far outweigh the negatives of not participating. Laboratories should be participating at least annually if possible. And there are many EQA providers, and I'll sort of highlight some of them at the back end of this talk. And there is EQA for most aspects of diagnostic services, but because there are a large number of genes tested, it's not possible to provide EQA for every single disease. So where there is no EQA available, laboratories are recommended to participate in some sort of interlaboratory comparison or exchange. But you are only as good as your last EQA. And every laboratory is capable of making a mistake, no matter how good. Next, please. So an interesting facet of providing EQA samples is that laboratories tend to treat an EQA sample differently from their diagnostic sample. What you really need to be doing is treating the sample as the same as a diagnostic sample. So following your usual diagnostic practice. There are obviously differences in terms of the samples that are provided by EQA providers. So many labs for genetics, many EQA providers for genetics would be sending out a sample that as closely mimics a real patient sample as possible, but generally it would be a DNA sample rather than blood. So we can't book EQA for the whole process, but certainly we can provide EQA for the bulk of the process. So we recommend also that you treat your sample in the normal way you would any diagnostic sample if possible. Your internal methods should be robust and reproducible, and you should start your analysis of the EQA samples as soon as possible as well. There is a tendency for laboratories participating in these activities to leave it to the last minute. As an EQA provider, we'd always insist that you contact us if you had any problems with the samples, and where possible, we're able to actually provide additional samples to help overcome any problems that you might have had with the initial sample. Next slide, please, Mark. So if you are looking using EQA samples, what we would recommend is you don't give your EQA analysis to your most experienced staff. This is an educational aspect, and you should be looking at the whole process. So it's a good idea at times to try and vary who staff who actually get the EQA sample to test. Don't involve the whole department in the processing and analysis of the sample. Remember, it should be mimicking a normal diagnostic sample and go down the normal workflow route. As I said before, don't leave your analysis to the last minute. Make sure that you always submit your reports in good time. You never know when a problem may occur, and you're not giving yourself. And if you don't give yourself sufficient time, you may not be able to submit reports in time to the EQA provider. And in many cases, that's deemed as an issue of poor performance on the basis of not submitting results in time. Next slide, please, Mark. So when it comes to reviewing the results from EQA, most providers only have two classifications, if you like, of performance. Either you are deemed a satisfactory performance or poor performance. Labs should be proud of achieving a satisfactory performance. The standards themselves for each EQA provider that define this cut-off point are usually quite tough, and so getting a satisfactory performance is something of note. You should address, where possible, any issues that have been raised by the EQA provider in their feedback report to you. This is particularly important if you've been deemed a poor performer on the basis of your results from the EQA. We would advocate that you review and audit your practice regularly as a review of the process, the laboratory process, and incorporate the EQA results into that. And it's good to benchmark your score against all the other laboratories. Is there any room for your improvement in your process? What can you do differently? And you can use the information provided by the laboratory feedback report from the lab, from the QA provider, to help you do this as well. Next, please, Mark. Next, please, Mark. Next, please. Not sure what's gone on there. Could I have the next, please, Mark? Oh, I seem to be going. I think it might have skipped a slide there. Could you go back one, please? So, learning and improving from EQA. Well, EQA is educational. That's the real key point here. It's not personal. So be objective about it. Don't expect full marks. Very few labs actually achieve full marks, but look at it as a benchmarking exercise, as I said before, comparing your performance to the other laboratories. Always review your EQA case results, particularly review the summary results that the provider has sent to you, and it's highly recommended that you share your EQA results with all the people working in your laboratory, so that you're all aware of how the laboratory is doing, and maybe address any of the issues, or talk as a group about any of the issues that have been raised from it. And many laboratories also use the EQA cases themselves for training of their staff. Okay, next, please. So the next 4 or 5 slides, I've just got some examples of why EQA is beneficial. And I'm going to take you through some examples from both the genotyping of a test through to the reporting of the test and the interpretation of the test result. So these slides are kindly provided to me by some of my collaborators. So this is a slide from the CF network, European CF network, and what it shows is the data from the CF scheme over a period of 10 years relating to genotyping, so the accuracy of genotyping essentially. And in the top line, you can see the percentage of laboratories that made a genotyping error. So back in 2000, when they launched the scheme, about 10% of the laboratories were making a genotyping error. And by 2010, you can see that that figure has dropped down from a high of 22.8% in 2002 down to about 2.8% of laboratories making a genotyping error. And that's also mirrored by the percentage of samples with genotypes incorrectly genotyped, and the number of alleles also incorrectly genotyped. So over time, what it's essentially showing is that the genotyping error rate is dropping with participation in the EQA scheme. Next, please, Mark. This is an example from the UK NEQUAS for molecular pathology, and it shows the first 2 years of a pilot EQA scheme for EGFR testing. So back in 2010, they launched the scheme, and I'm not sure how many participant labs there were, about 30 if I remember rightly, and nearly a quarter of those laboratories made a genotyping error, made genotyping errors in the scheme. They only send out 3 samples, so it's a very high number of genotyping errors. The next year, they split the scheme into 3 distributions, each with 3 samples. So that's 3 times as many samples split across the year. And you can see that in each case, the genotyping error rate dropped dramatically as a consequence of the feedback from the EQA provider about the genotyping assays and the performance of the laboratories. So it varied in that case between 9% and 5%, but still considerably better than 25% that it was in the first run the year before. Next, please, Mark. If we look at the reporting, this is an example from the CEQA, the Cytogenetics EQA, European Cytogenetics EQA, and EMQN, who run a EQA scheme for constitutional microarray testing, array CGH. And on the top figure, you can see the results of this particular lab. In 2009, sent a report for the one sample that was 5 pages long. And best practice dictates that we try and produce a report that's clear and succinct, and preferably no more than 1 page. I know it's difficult with particularly things like array CGH and maybe next-gen sequencing, but clear and succinct is the way to be going, and it's certainly written into the relevant best practice guidelines and the OECD guidelines that we do this. So in 2009, it provided a report for this one sample we sent that was 5 pages long. 2 years later, after having provided every laboratory with sort of example reports of how they could present their results, the laboratory participated in the scheme again, and you can see that their report was essentially back down to 1 page. So they've compressed all that information into 1 page, with a second subsidiary page with some sort of additional information that just about the testing process itself. Interestingly enough, they've gone from having 3 signatures on the report to 6 signatures on the report. And unfortunately for the laboratory themselves, they also got the genotype wrong. So despite having checked it 6 times, it was wrong. That's just an aside. Next slide, please, Mark. If we look at report content, the actual content of the report itself, the aspects that the quality assurance scheme has deemed important for that result, and this is all based on the best practice guidelines itself. This is an example from the CF scheme again, and it shows the improvement in interpretation of cystic fibrosis reports over a longitudinal survey. So this is 5 years worth of data, and 2 example cases where they've looked at the reports that have a sort of common theme, if you like. So, for example, a confirmation of CF genotype. They include qualification of genotype. Next, please, Mark. And in all cases, except for one, they observed a positive effect on the number of previous participations, on the presence of the specific elements we talked about, that are outlined in the graphs above, except for the confirmation of CF carrier status in Figure 2. So what that's saying is basically, if you participate regularly and listen to the feedback from the EQA providers, performance of laboratories improved over this 4 or 5 year time span in terms of the report content, the aspects they were assessing the labs against. Next slide, please, Mark. If we look at the interpretation of the results, this is again, it's from the CF scheme. If you press next one, please, Mark. They also observed a positive effect from the number of previous participations on the score from the laboratory. So each laboratory is given a numerical score for their performance. Essentially 2 marks is a completely correct interpretation with all the aspects that the assessment team have deemed important in that report. 0 be a report that contains no interpretation whatsoever. And you can see on the top line, the top line, the grey line, represents the labs that have participated in the scheme previously, and you can see their performance improves over time when compared to the whole group of participants and those labs that only participated once, which is the bottom grey line. Okay, so that demonstrates the importance of participating regularly. Another example and final example is in the issues of mutation nomenclature. This is taken from the EMQN scheme for congenital adrenal hyperplasia. And the question is raised, are we really testing the same mutation? So in 2008, one sample had 28 different nomenclature descriptions from 38 participants, and I'm just giving you a few examples here. So 38 labs, 28 of them gave different descriptions of essentially the same mutation. And over time, so this scheme's been running since 2008, we've seen a year-on-year improvement in this, such that applying these nomenclature rules, so the EMQN recommends the use of HGVS nomenclature. Applying those nomenclature rules has reduced the variation in reporting of genotypes significantly. So in 2011, we pretty much have consistency across all laboratories for how they interpret and report a particular mutation. Next, please, Mark. So if we're looking at what I've done in the next few slides is just try to pull together sort of examples of providers for the different areas of EQA that I'm aware of. So if we look at molecular genetics EQA, I guess the 4 largest providers for molecular genetics are the EMQN itself, which has over 1,200 laboratories from around the world participating. And the EMQN assesses not just the genotyping but also the results interpretation, has no restrictions on participation. The UK NEQUAS is essentially the model that EMQN started from originally, but it's predominantly UK-based, but not entirely. Again, assesses the whole laboratory process for the genotyping interpretation, but there are some restrictions on participation depending on the scheme you look at. There's a European CF network scheme, which obviously just tests CF. Again, uses a similar model to the EMQN and UK NEQUAS, so no restriction on participation. And then for those of you based in the States, you'll be much more familiar with CAP, which has a similar sort of basis to all the other providers, but it's mainly US-based. Only assesses genotyping, and I think that's a crucial point. I think in these molecular genetic tests, the interpretation is just as important as the genotyping, but has no restriction on participation. But they're the 4 biggest. But if you put next, please, Mark. But there are many other providers also for molecular genetics, and I've just listed them all here. But if you go to the next, please, Mark, to the web link below, which is a link to the EuroGenTest website, it'll give you a table that just breaks down all of these providers by the diseases and EQA schemes that they actually provide. So it's a much more detailed overview of it. Next, please, Mark. And just to give you a feel for the sort of things that these providers offer, so for EMQN, we offer a range of different monogenic diseases, as well as some technical EQA schemes, record karyotyping, and then 2 sequencing schemes, one for Sanger and a pilot for next-gen sequencing. And more increasingly, we're moving into the field of molecular pathology with EGFR and BRAF testing already being offered, and several other schemes being in development as well. Next, please, Mark. For cytogenetics, it's pretty much the same. It's that the EMQN is replaced by the CEQA, which is the European cytogenetics EQA. But again, the same remit as EMQN, sort of pan-European, maybe worldwide, no restriction on participation. Again, the UK NEQUAS scheme is exactly the same, but predominantly UK-based, but the model is the same. And then we have CAP as well. And a few other providers. Mark, next, please. And again, you can find more information from the next link, which is the EuroGenTest website. Next, please, Mark. For biochemical genetics, I'm only really aware of one provider for that, and that's the EMQN network. I'm not going to try and pronounce the acronym because I can never remember it. But if you're look up for the EMQN website, it will give you a detailed description of the EQA schemes they run. They're a worldwide provider of EQA, and as far as I'm aware, there's no restriction on participation of laboratories in that network. Next, please. Pharmacogenetics. There are a number of providers for pharmacogenetics EQA schemes. And again, the Eurogentest website has got a list of this. I think it's probably, I think there are more now than there are currently on the list there, but it's reasonably comprehensive. Next, please. And finally, for molecular pathology, again, we have the EMQN schemes, which I've already described. The UK NECWAS also does a number of molecular pathology schemes. The European Society of Pathology also provides schemes for KRAS and is also this year is providing a pilot scheme for lung cancer that includes not just EGFR testing but ALK testing as well. I'm very aware of the schemes obviously that are going on in CAP as well for this area. There's a lot of overlap between the providers so far, but there are also many national EU schemes provided out of Netherlands, Germany, Austria, Italy and more, I think, as far as I think there's a French one as well. So there's plenty going on in terms of molecular pathology EQA, but what there isn't is an easy access to that information from any one central portal. So you have to go to the providers or do a Google or web search to try and find out more information about what's provided. Okay, next please, Mark. So finally, just a few useful links to give you some more information, really just to the main EQA providers that I've highlighted so far, the European ones. And the bottom link is the Eurogentest website, which, if you haven't checked it out, would be worth looking if you want to find out more information about EQA, but also laboratory testing and standardization, best practice, etc., around Europe. Okay, that's it, I think.
Great. Well, thank you very much, Simon. Lots of great information there, and lots of questions that are coming through. So we've got time for some questions. The first question, actually the first two are kind of related, and they're for you, Simon. How can you manage across international borders, and how do you manage the regulatory environment in each of the countries? Can you address that?
Is that in the context of EQA?
That's right, yeah.
Well, for us, I must admit, we never really had a problem. In fact, we never even thought of it as an issue. I mean, our schemes are provided to 64 countries around the world. So there's because we're sending out usually a genomic DNA sample, we don't have any problems with sending the samples through via courier. So we don't send out our blood sample because of the problems associated with sending out those sort of delicate samples. So our main analyte is DNA. So I must admit we've never had a problem with the process of sending samples, and so as far as I'm aware, never really any problems with the regulatory environment at all. I mean, the issue really for the regulation is the laboratory themselves. So are they actually participating there as activities? But for us as an EQA provider, that's not been a problem.
Okay, great. Here's a question. How important do you feel the ISO 15189 is for labs? I personally feel it's very important. It gives the labs a structure to base their services around. It defines a sort of a quality management system which they can work to. And I feel quite strongly that the laboratory should be working to ISO 189 or an equivalent accreditation standard. I understand that some of the arguments about the cost involved to it, and it's not insignificant, the amount of work and effort and cost that's involved in setting up a laboratory to those accreditation standards. But I feel strongly that the benefits far outweigh the negatives. Great, thank you. Here's a question for Maria. Maria, you mentioned group purchasing. Can you explain what specifically group purchasing you do with the network?
Well, basically, we are starting to manage that for diagnostic with different diagnostic companies, group purchasing and reagents. Say the major reagents that you use in a lab, say QIAGEN, Roche, Abbott, the different brands. So what we've done is to place a survey in Connect, in the farm section where it says group purchasing, And we ask the different participants and the different users to send us their insights and their preferences in the different reagents that they most commonly use and the most expensive ones. So it'll be great that anyone posts their preferences so that we are in that position to negotiate those prices for anyone. The idea would be to have a tool inside Connect where you can place the orders and get that discount, or to negotiate a rate with a code for each of the members in Connect when they buy that reagent or that equipment, whatever it is.
Okay, thank you. This is a related question, Maria. How do you use the survey information you showed within the network?
Well, there are two different sections regarding the information that we retrieve. One is the public information that we show, and it's the one that any user can decide to show. I mean, it's publicly shown, and they decide what's the information they want it to be disclosed. And then there is another survey, and there is another section which is the information that we retrieve in the survey, and that's confidential. So we are not showing that information. So basically, each user decides what's the information that they want to be publicly disclosed.
Okay, perfect. Another question for Simon. Simon, how is poor performance addressed in terms of recommendations? Do you provide recommendations if there's poor performance?
That's a very good question. Poor performance is a bit of a mixed bag depending on which regulatory environment you're in. So for some countries where there's a good regulatory system set up, for example, like the UK, there is a sort of national mechanism for reporting poor performance to the relevant bodies. But for others, many countries, obviously, there is nothing like that. So it's a bit more difficult to deal with. But essentially, we deal with the process of poor performance. We set poor performance criteria. If laboratories don't meet the poor performance criteria, we would write to them to inform them that they haven't met the poor performance criteria. And then we open an iterative dialogue with them where we ask them to discuss the problems with the EMQN and offer them help and education where possible, or access to resources like other laboratories who are doing the testing to allow them to improve their performance. In some cases, again, depending on where they're coming from, we might also send them additional samples to test, so if like an extra round of EQA to establish whether they have dealt with their issues of poor performance in the first round. But that's limited, and it really tends to be, for example, where the UK, where we're able to, there's an obligation to really go further with dealing with the poor performance. But in many cases, there's not a lot we can do apart from offer them advice and help and assistance to try and improve their performance. It's really up to the laboratories in many countries to do the work of improving their performance subsequent to EQA.
Great, thank you. A follow-up question to that: this is for Simon, are there specific tests where you see more importance for EQA testing?
Yes. So I think for the established testing, like lots of the single gene disorders, which have been the bulk of our business over time, generally, the chance of making an error, the likelihood of a laboratory making an error, is down at the 2 to 5% rate, which is still quite high, but we have a lot of new labs coming in all the time. So if you've been doing EQA for a long time, as the CF data shows, you're less likely to make a genotyping error. However, what we are seeing is for the newer tests related to personalised medicine, for example, the EGFR and the BRAF test and the molecular pathology test, there is an intrinsically much higher error rate, which I think is caused by a number of factors, but partly because of the type of sample you're dealing with, and it's a more tricky and difficult sample to deal with, but also because I think laboratories are able to buy these tests as kits off the shelf or develop them laboratory LDTs, But what they're not doing well is validating their assays properly and designing their assays properly and ensuring that they're performing properly before they actually use them in service. I think there's too much of a tendency to be able to introduce them very easily because they're predominantly kit-based things, and not enough of an understanding of the challenges of the analytes they're working with, or the disease itself, particularly the cancer itself. And I think you really need to understand that properly to be able to interpret the results properly and get the best out of it. And also, there's a real significant lack of reference materials available for labs to actually validate their tests properly. And again, it's all part and parcel of this problem with validation itself.
Great, thank you. We've got time for a couple more questions. This one is for Tiffany. Tiffany, you mentioned that pharmaceutical diagnostic companies and laboratories need to work together. Can you expand more on how laboratories can help pharmaceutical and diagnostic companies in personalized medicine?
Absolutely. The laboratories are in such close relationship and contact with the physicians out in the community. The ones that are seeing the patients are prescribing both the diagnostic and then, if appropriate, the therapeutic. And the laboratories have a real significant role in being able to provide education to the physicians, to be able to understand some of their needs, and then to be able to communicate that back to the pharmaceutical company, whether it be in better knowledge tools that may be necessary. And that's really part of, as I talked about earlier, the whole premise of Labceutics is to be able to help to facilitate that connection between those stakeholders.
Great, thanks, Tiffany. And I think we have time for one last question. If we didn't answer your question, we do have them recorded here, and what we will do is we can reach out to you personally by email with some answers. But we will answer this last question. This is for Simon. Simon, are the results made public in any medium in terms of the testing that you do? So I'm guessing they're asking, are they made public to the general public?
So the participation of the lab is made public, and EMQN and SQL and IPTS network and other providers, European providers, feed back directly to the Orphanet website, so that you can go on to Orphanet and look at the laboratory.
Great. Simon, could you actually just repeat that? I think we're having a little trouble hearing you.
Sorry, Mark. Sorry.
Yeah, that's perfect.
The participation of the labs is publicly available. So a lot of the providers, the EMQN, SQL, UCF network, feed in directly to the Orphanet website. And on there, you'll be able to, if you logged into it and look for the laboratory information, you'll be able to see whether a laboratory is a participant in EQA and what schemes they participated in and when they participated. But the performance of the laboratory is not published. However, each laboratory that participates in the EMQN gets a certificate, and on that certificate is the performance of the laboratory. So if you are purchasing services from a laboratory or looking at trying to use a laboratory for setting up a test, I would recommend that you ask them for their EQA certificate to see their performance scores.
Great. Well, thanks so much, Simon, Maria, and Tiffany, for a great discussion and panel today. I know I learned a lot, and there was some great information that was shared with everyone. And thanks to you and the audience for taking the time. As I said, if you have a question, please follow up. You can follow up directly with Susanna Lobo. Her information is on the screen, and we will be happy to answer any questions that you have. So with that, I will wish you all a great rest of your day. Bye-bye.